Hello, my friends. My name is Joshua Mugabi. I welcome you to Biology King. And in this session, we are going to look at origin of the cell. Describe the four processes needed for the spontaneous origin of life on Earth. There was non-living synthesis of simple organic molecules that was from primordialin organic molecules. These simple organic molecules became assembled into more complex polymers. Certain polymers formed the capacity to self-replicate, that is, enabling inheritance. These molecules became packed into membranes with a distinct internal chemistry, or what we refer to as protobionts. Outline with the aid of the diagram the Miller-Urey experiment into the origins of organic compounds. Water was boiled to vapor to reflect the high temperatures common to Earth's original conditions. The vapor was mixed with a variety of gases to create a reducing atmosphere where there is no oxygen. This mixture was then exposed to an electric discharge, simulating effects of lighting as energy source. The mixture was then allowed to cool, concentrating components, and left for a period of approximately 1 week. Later, the condensed mixture was analyzed and found to contain traces of simple organic molecules. Identify the conditions needed for a non-living synthesis of organic matter and potential locations. Conditions: reducing atmosphere, that is, no oxygen, and high temperatures or electrical discharges. Locations: we have volcanoes, hydrothermal vents, and meteors, or panspermia. Outline the evidence from past experiments that demonstrates that abiogenesis no longer occurs. Broth stored in vessels with long tubing, that is, Swanneck ducts, that can prevent passage of dust particles. The broth were boiled to kill any microorganism present in the growth medium, or what we call sterilization. Growth only occurred in the broth if the flask was broken open exposing the content to contaminants. From this, it was concluded that bacterial growth came from contaminants and did not spontaneously occur. Outline two properties of RNA that would have allowed it to play a role in the formation of life. Can self-replicate, although superseded by DNA due to the double strand. Secondly, can act as catalyst, although superseded by proteins due to greater variability in subunits. Describe the process of endosymbiosis and how it led to the evolution of eukaryotic cells. An endosymbiote is a cell which lives inside another cell with mutual benefit. Eukaryotic cells are believed to have evolved from early prokaryotes that were engulfed by phagocytosis. Over generations, the engulfed cells lost its independent utility and became a supplemental organelle. The engulfed cell remained undigested as it contributed new functionality to the engulfing cell. Outline the evidence supporting the theory of endosymbiosis. We have the components and the evidence. Membranes. Chloroplast and mitochondria have double membranes. Antibiotics. Chloroplasts and mitochondria are susceptible to certain antibiotics. Division. Chloroplasts and mitochondria divide by a fission-like process. DNA. Chloroplast and mitochondria have unicircular DNA. Ribosomes. Chloroplast and mitochondria have 70S ribosomes. Hello friends, thank you very much for participating in Biology King. I encourage you to subscribe.